Welcome back. This is lecture 4 in our Dynamics on Network Modeling class. Today, I'm going to be talking about steady state, enzyme kinetics, Michaelis-Menten, and Hill rate laws, getting started on that, and a little bit on parameter scan techniques. And I'm going to start out early in the class by asking people to give short presentations of the work that they did preparing for the little projects. So we're going to start out with that student projects, and I'd like people, if possible, to screen share and present from their laptops. It's the easiest way to do it. And then we'll talk about chemical equilibria. We'll review some of the concepts from the previous couple of weeks based on what people were doing in the homeworks. And then we will talk about enzyme kinetics. It looks like a lot of exercises, but actually most of these exercises involve changing one value. You have a basic simulation and you change one number and you run it. So the exercises take about 10 seconds each. Once you've got the first one maybe takes a few minutes. And then once you've got that, they're pretty fast. And I also have all of them uploaded to the student materials. And I just checked that works, so they should all be there. And I'll talk a little bit about, there are a number of ways of doing parameter scans, some of which are more convenient than others. NumPy has two array manipulation operators called VStack and HStack, which are not the most enjoyable things to use, but are a good way of assembling information. So we'll see how far we get on that. William helpfully pointed out that somehow nanoHUB broke the nanoHUB installation of Tellurium. In fact, nanoHUB seems to have broken all of the Jupyter notebooks for all things the Jupyter notebooks on nanoHUB. The issue is that the directory, the right directory that should be your user home directory now points to a session directory, and you can't move out of that session directory to save things into your home directory. And when you then leave the session directory is eliminated. So it's not the most convenient thing in the world, but if you save locally, you do your save and checkpoint, and then you do download as notebook, it will save everything to your local computer. And then when you hit open, there is an upload button under the open that will allow you to pull it back from your local computer. So provided that you remember to download it to save locally instead of on the server, it's not too bad. And I did all the exercises to check that it wasn't too bad. I did all the exercises for today that way. And I'd say it added an extra 5 minutes or 10 minutes to my afternoon, but it's not terrible. It's only maybe 15, 20 seconds extra each way. The key thing is if you're going to use it, you've got to remember to do it. So I'd like to start out by having people talk a little bit about their paper lists. I've given you these series of assignments of looking at papers, thinking about things in the hope that you find something that's interesting to you. The goal is that you should feel free to change your mind about what it is you want to do between now and whenever you decide to start on it. And so the goal of all these little assignments is to try to have people explore ideas which is enough that you can think about whether you're interested or not without making it so burdensome that you say that you're a sort of sunk cost fallacy where you say, I've worked so much on this project, I have to continue with it even though I hate it. Try to avoid that situation. Okay.
So for the project, for the papers that I was looking through, hi, by the way, my name is Ibraheem. I'm a second-year PhD student in the ISE department, and my focus is in bioengineering. And I guess some preface. So recently I've been taking some ecology-related courses. So I've been pretty interested in ecological predator-prey modeling. So I kind of was digging a little bit into modeling predator-prey interactions for, I guess, what I was interested in for the project. So basically some background so I picked like 3-ish papers, but one of them I'm kind of mainly going into. The other 2 are kind of just tagging along as examples of other predator-prey models. So basically some background is basically there are a lot of different ways to express or model predator-prey interactions, each varying in complexity and in their specific predator-prey dynamics. So one of the papers, so basically in most situations where entities interact by sharing limited resources, controlling population density is important to maintaining ecosystem stability. And in this, so there are some predator-prey interaction models basically that it's important to account for harvesting and consuming resources. So predator survival is related to its ability to consume resources as well as prey survival. So one of the important models for that is called the Rosenwig-MacArthur predator-prey model, and it's characterized by its ability to do this kind of modeling, harvesting, consuming resources, as well as its ability to incorporate a delayed conversion of prey into resources. So this is, I guess, it's useful and interesting because it can more, I guess, it's able to dynamically or just better display the dynamics of predator-prey interactions, I guess, more interestingly. So if we look at some of the initial equations for this, here we can see on the right is the total equation. On the left is kind of, I broke it up into some smaller parts. Basically, what happens here is on the right is the rate of change of the prey, and this is dependent on kind of 2 sections. One is P times 1 minus P over K, and the other is Q over A plus P. And in this scenario, basically this P times 1 minus P over K is, so maybe I'll go over the letters quickly. So P, big P is the population density of prey. K is the carrying capacity of the ecosystem. Q is the population density of the predator, and alpha or A is it's quantified as the conversion rate of turning prey into usable resources. So basically the rate of change of prey is a multiple or related to both the prey. So basically the first section of this basically represents saying that prey the rate of change of prey will increase if there is less prey in the system. Basically saying that if there's less prey, there are more resources for the prey to obtain, therefore you will get more prey in the system. So that's this first section. The second section is basically saying the rate of change of prey will decrease if the predators are able to convert prey into resources efficiently. So just simply put, less prey and less predators and worse conversion efficiency will lead to greater prey concentrations. So predators, on the other hand, is basically related to the amount of predators. Beta is just the it's just the intrinsic growth factor of predators. So predator rate of change is related to the growth rate of the predators. themselves. And also this is the predators, how the predators version of obtaining resources from the conversion rate. So here, instead of it being minus related to the predators concentration, instead it is positive in relation to the prey's conversion concentration. Basically, we're just saying that if the predator is able to convert prey into resources efficiently, it will also increase the rate. This T minus tilde or tau, I guess, is just the accounting for a time delay in the system, and that's the nature of the specific system, is that it incorporates this time delay. And this time delay is interesting because it helps us to give us, I guess, a more dynamic system. So here, if we look at in the paper, they basically take that initial model and give it initial conditions, or I guess assumptions of the model that they will use to basically define parameters for the system, so we can get, I guess, a more lifelike system. So basically, it's the differential equations are basically the same. The only difference is they add this theta and this c in both the predator and the prey and the predator functions, and basically those are just the decay factors per unit of time for the prey and the predators. So it's just the initial equation with the addition of both the theta and a c. Some more parameters are basically the k, as in the carrying capacity, can only be equal to the sum of the prey population and the predator population, assuming that both the predator and the prey population is less than the carrying capacity. Also, the initial concentration or the initial population of predators and preys is greater than 0, and also their growth factors and the conversion rate of the system is greater than 0. And with this, they basically were saying that this effectively will model predator-prey interactions with this kind of resource management kind of framework in mind. So some additional models. These models weren't in the paper I'm kind of talking about, but these are some of the other models that I would kind of like to explore. There's this anti-predator response model. Basically, it divides, without going into too much detail, basically divides predators and preys into adult predators and adult preys. And adult prey, I think it's this one, basically has anti-predator responses. So if the juvenile prey can develop into an adult prey, it will develop some anti-predator responses, and that will modify the system. Another one is this ratio-dependent system with predator harvesting. It's quite similar to the one we were looking at, just with a couple more parameters. And I think this one would also be pretty interesting to look into. But basically, just going back to the first paper, here's just some examples of their simulations. This example that basically the rest of the paper was discussing kind of modifying different parameters to see what happens. So here on the right is kind of a generic system with a time delay of 0. So as in the predators can harvest, so the predators can harvest prey and obtain resources at an equivalent at the same time as the prey. So here we can see kind of just a normal population diagram where the prey can increase and so can the predator population. But if we notice, if we increase or if we provide some time delay, the ability for predators to obtain resources in a timely fashion decreases rapidly. So the predators actually end up going extinct. And the preys have a far easier time existing in the ecosystem. So that's, I guess, interesting. Which kind of resulted in me wondering why we even would want a time delay in the system if it seems problematic. But I guess what they realized was it was interesting because if you have a time delay, this is an example with a time delay. So this is kind of, if you modify other characteristics, you can alternate between a kind of a random, you get this this random type behavior in the system, and if you modify different parameters, you can get back to a stable system. So that's, I guess, without the time delay, you don't get this kind of randomness effect. But if you do implement a time delay, you get this randomness effect, which you can solve by modifying other parameters of the equation. So basically, this main paper kind of applied this time delay effect in predator-prey populations to network solutions. They referenced how these dynamic characteristics of the system are applicable to networks and packet transfer. So that's kind of how they were using this, which is kind of why they wanted this time delay. But more importantly for me, I think it's a kind of a proof of concept for modeling predator-prey interactions. And it kind of provides a framework for modeling and simulating other predator-prey model types, which I think would be a pretty useful thing to do. These are just some of the references I had, and that's basically the presentation.
Okay, thank you very much. I was just going to type in the chat. People shouldn't feel that you have to be this detailed in your presentations. That's great, but I wasn't expecting that level of detail for a couple more weeks. So you've thought about that a lot. I mean, one issue we're going to face with antimony is that you can't write delay equations in antimony. There are ways of approximating them. But some of the equations you showed don't have delays, so those are easy. The delay ones are a little bit harder. We could certainly do it, but it's an interesting case. The Rosenzweig-MacArthur model is a very, very old model. I mean, it's 75 years or more old, and it's interesting to see that it's still being used and extended. Some of those basic ecological models have had a lot of life to them. So thank you very much. That was great. So there was a question which was, what does the delay simulate in the real world?
So well, in the context of this one, I'm under the impression that it's related to giving the, I guess in this specific scenario of predator-prey interactions, it's kind of just giving the prey a head start in obtaining the resources or in developing in the system, and the prey kind of has to catch up. I don't exactly know what it would be in just general broad terms for network modeling.
Well, but here specifically, if I'm modeling, you could think about the population density or the number of animals of each species. Reproduction takes some amount of time. So if I eat X kilograms of prey, I don't immediately have another animal. I have some gestation time, and the baby animal can't probably hunt and become another predator for some amount of time. So typically the population would represent the number of adults of a given species, and therefore you have a lag, which is how long it takes from the time you eat to the time you get another animal. And so those delays can be quite substantial. And actually the fact that models without delays sometimes work pretty well is a bit surprising. You might expect instead that you have to have the delays to have things work.
Okay. So I was looking at network analysis for invasive species dispersal, and then I found a very extensive thing that was looking at how you would analyze the networks. And there's just two main like things you're looking at is the human interaction mediation for that, and then also like the probability of it spreading within the specific ecosystems it's in. And I wanted to look closer and see if like it's possible to create a model to a specific like ecosystem that would be easier. And so I chose stonewort, which aquatic invasive plant that just spreads a lot in lakes. And I tried to find like just a small collection of lakes or something that has extensive models of like human interaction throughout the lakes. And I found one in Minnesota in Stern County, and they use a lot of like statistical data to analyze the probability of one boater from going from one lake to another from there. And it's looking purely just at like ability to move from one lake to the other. Like I said, much simpler.
That's actually a pretty sophisticated model. I think could be quite interesting.
Well, yeah, there's also another more sophisticated, there's a more sophisticated model I found that was actually using just like network analysis of the entire like statistical data they had to look at all lakes in Minnesota, but I figured that would be too complicated to analyze, so I just ignored that.
I didn't understand.
You didn't understand?
No.
Okay, that's fine. So invasive species dispersal of like a specific aquatic plant, looking at each node as represented as a lake. And so there will be two possible values for each node, specifically whether the amount of aquatic species that are in the lake, and then the edges between each lake will be the amount of human interaction between lakes. That would be like analyzing the likelihood of dispersal from the aquatic species from one lake to the other. Yeah, from the ballast from boats when they're moving from one lake to the other. The lakes are not connected in this case.
So how can we transport by boat?
So the boats will have ballast. Yeah, okay. So boats will have ballast water collected just for like having correct buoyancy levels. And so the main amount of invasive species spread in aquatic lakes are from that ballast collected and then not being like disposed of before you go to a different lake. Or it'll connect on like the rudders and it's not properly cleaned or something like that.
So probably the distance between lakes will matter?
Not the distance between the lakes, but the amount of movement from one lake to the other from boaters. So the model already existing is talking about like movement, human interaction within a region, but there's nothing including like the specific invasive species within that.
Right. The trick here will be representing the nodes and
Yeah.
the links, because you've got a lot of nodes, and so then this is. But it's certainly possible. You have some densities on each node. This may be a case where antimony isn't the best way to do it. I mean, Python dictionaries will be a great way to do it. Whether you want to use antimony to solve the network dynamics is another question, but it's a great problem. Right. So Pedro says, well, you could simulate what happens when you change the number of nodes in the connectivity. And that's certainly the kind of thing you might very well want to do with this kind of model. I'm not sure antimony is the best way to do it. I mean, I think, but it's certainly appropriate for the class, and certainly it's not hard to write a Python-based model of this kind, where you have a network, a directed graph network, and then you have a set of species living at each node with a probability of infection. So I think a couple of people were interested in infection models of one sort or another, and this is a nice example of that. And so I tend to always think about things that we could do with antimony, because that's what we teach in the class. But there's no reason that you can't do a model that uses other methods. And this is quite an interesting one. It's very much in the style of the work that was done by by Alessandro Vespignani on influenza and COVID infection, and by Alessandro Flamini, who's also here now.
So I'm William. I'm a computer science student, so I don't know much about biology. So I sort of was just going around looking for things that interested me. So first topic that interested me was cancer. Okay, so I came across this paper on how they basically simulated, they simulated and understood the epidermal growth factor receptor, and I'm just reading this because I can't remember it. Type 1 insulin-like growth factor receptor pathways in non-small cell lung cancer. So this model basically, I don't know why I'm blanking. Sorry. So on the left, there's a graph of the three, basically the three like settings. One is when EFGR, which is like what I just said, epidermal growth factor receptor, and the other one, insulin growth factor receptors, when they both start at a, this is kind of confusing. They're both down expressed, so they're both expressed at a lesser frequency or activation, and then they're overexpressed, and they're just in the wild. I don't know wild is with an E, but so what they did was they modeled different levels of EFGR and IGF-1R, and they basically were looking for the amount of you can't see it on here. Wait, they were looking for the amount of I think ERK pathways or ERK, which then the more active an ERK pathway is, this will result in more growth of cancer cells and survival of cancer cells. So we see on the left graph, the left bottom, the pink graph, when both of the receptor pathways are co-expressed, this will result in the greatest amount of ERK, ERK pathway activation, which will, so this is what they explored. They found that when both of them are expressed, cancer is more likely because of the ERK pathway increase in activation. I think those are the right terminologies. I'm not sure. But I was interested in, or I want to further explore, exploring other like related pathways and seeing that those impacts on cancer. But this paper I was interested in, but it's not my favorite. This is a cell budding yeast. I think is just normal. It's yeast that's just growing. So basically, since yeast is a very well-studied and well-known like molecule organism, it's hard to model, which was very interesting to me. The more you know about something, the harder it is to model. But so on the left, this graph, this complicated sort of mess, is the like current consensus of all scientists on how to show the cell cycle of budding yeast. So what these researchers did is they created a mathematical model they try to replicate this model using ordinary differential equations, and test it on 130 plus mutants of yeast, and to see if this model stands true for all of the mutants. And it mostly did. 120 of them stood true, and I now want to investigate the ones that did not work out by tweaking the model, seeing what happens, stuff like that. And then this was the last one. Okay, I honestly I haven't tried to understand the pictures yet, but this one was by far the most interesting to me because I did not know about quiescence, which is, so basically this this whole paper is about how when a cell there's DNA damage in a human cell, how the cell responds and makes a decision on whether to continue the cell cycle by splitting or undergo the process of quiescence, which basically is to stop, to stop proliferation, to stop splitting. It's just going to, there was a phrase, but it's just going to stop. And these researchers wanted to find out, okay, what is the like most important points in the cell cycle? I think that I'm not 100% sure. And so the scientists wanted to figure out, okay, where are the most hang on, I have some of this written down. Okay, yeah. So the model that they created basically tried to replicate the decision-making process of these cells. And they figured out that at the restriction point, which I don't know what that is, and the G1/transition phase points, those are the most important 2 points when it comes to decision-making in a cell, whether to proliferate or go into the process of quiescence, thereby, and this is very important because if these 2 points are like disrupted or anything, this could lead to more mutations and cancer and stuff like that, bad stuff that you don't want. So yeah, that's what I did, and I GIF. So yeah.
So you have two papers on cell cycle that you're looking at. It's
Yeah.
interesting that both of those, you see a lot of shared molecular species. I was saying the other day, when you're looking at gene names, there are a lot of names, and so it's not reasonable to assume that you learn all of them at once. But for example, if you see 2 papers and half the genes are the same in both papers, then those are probably the ones you want to start paying attention to. And so you'll see a lot of things that are called CDC, which are or CDK, cyclin-dependent kinases, cyclins, because they're related to the cell cycle. So when you see CD, for example, or CDK, those are names that are typically associated with cell cycle. And then in that second one you showed, there were also things like P53, which is famously associated with programmed cell death with apoptosis, but also can be connected to cell cycle and proliferation. I think you had P21 as well, which is another molecule that has these helps cells make these choices between survival or stasis, or quiescence, proliferation, and then death. And so, as you point out, from the point of view of an organism, multicellular organism at least, if there's something that goes wrong with a cell, you'd rather have it die or at least not do any, not proliferate. The thing you really don't want is for the cells to proliferate when they're not supposed to. And because cell replication is so complicated, involves so many steps, there are a lot of what are called checkpoints, which are places in the cell cycle where the cell essentially checks to make sure, for example, are all the chromosomes replicated properly? Are they aligned properly? Are they divided between the 2 daughter cells properly? And if the answer is no, then there are mechanisms either to correct, to fix the problem, or to say, I can't fix the problem, I'm just going to stop doing anything, or to say, I really can't fix the problem, I'm going to die, I'm going to kill myself. The restriction point and the other checkpoints are related to these decisions about whether you feel safe to go. It's like a pre-flight check for an airplane. If there's a problem with the engine, you don't want to take off. If there's a problem with the toilet, maybe they'll take off anyway. And so there are these relatively complicated mechanisms that have lots of levels of backup, trying to ensure that you don't have disastrous consequences. And so they're both very interesting problems. Yeast as single-celled organisms can tolerate a higher rate of error, but yeast as well, if you're producing a lot of offspring that are defective, then that's evolutionarily unfavorable as well, because you're wasting resources. And so yeast have most of the checkpoints that you find in multicellular organisms already. It's a pity. We had a professor here who was one of the world's experts in the biology department, Joe Pomeraning, who did incredible work. Sorry? Yeah, there are multiple. Yeah, so there are a bunch of people who worked on this problem here at IU. So it's a very topical problem, and people keep discovering yet more checkpoints and ways of constraining these things. One model that we may well do later in class, or we have in some years, is a very simple model relatively simple model of cell cycle due to Tyson, which is about 25, 30 years old now, but that models the main cyclins and their interactions. Because when I talked a little bit about gene regulatory networks. So one pattern that you get typically in cell cycle is that gene 1 turns on gene 2. Gene 2 turns on gene 3. Gene 3 turns off gene 1. And that's how you get a directional movement through the cell cycle. Gene 1 turns gene 2 on. Gene 2 turns gene 3 on. 3 turns 1 off. 3 turns 4 on. 4 turns 2 off. And so that way you can get a directional. If you just have 1 and 2, 1 turns off 2 and 2 turns off 1, then you can't get directional movement through the cycle. You have to have the extra element of the phase to make that work. And so that's a pretty classic pattern of behavior. So it's pretty interesting. That's a great one. They're all good. They're all really nice projects.
Yeah, my name's Justin. I'm a PhD candidate in the Genome Cell and Developmental Biology program. I found this paper that I found very interesting, although it doesn't directly touch my dissertation project. I thought, being a developmental geneticist, it was still pretty interesting. So okay. So I'm interested in cell fate decisions, or this idea of cellular differentiation, which is the process by which a progenitor cell, seen here as a myeloid stem cell, which is a multipotent stem cell, meaning that it can take on very different traits or fates. And all of this depends on induction of GRNs through different transcription factors. So for example, the transcription factor GATA1 will induce the myeloid stem cell to take a more red blood fate, where the PU.1 will make the myeloid stem cell go towards a more white blood cell fate. And currently, it's believed that there's an antagonism between these two transcription factors. So induction of GATA1 will try to repress PU.1, and vice versa. So PU1 express, it will try to downregulate GATA1. So there's this antagonism between these two transcription factors, and I'm kind of interested in trying to model how this works. So in the paper, they use this data-driven modeling method called gene circuits. I'm not directly familiar with this model, but apparently it not only is able to give you the topology of the GRN, but can also give you the type, whether it's the activator, inhibitor, or so on, just given quantitative data through either RNA-seq or ChIP-seq, different types of experimental data. And using this model, they used 12 genes to train the model, and their predicted model was able to find both knockouts, or predict knockouts, knockdowns, and overexpressions of these key transcription factors. So yeah, I would like to do something similar to this.
Yeah, I
Thank
think
you.
that's a great topic. That particular model structure is typical of how people do Boolean network modeling, which again isn't what we focus on in this class. There's nothing wrong with it. It's a great topic. Now, the only reason I tried to encourage people to do things that we can do with antibodies was that's so much we spend so much of the class doing. But of course, if the network you wanted to work with isn't one that's susceptible to that, that's fine. I'm pretty sure that there are going to be interesting hematopoietic differentiation models, which will be available actually in SBML. I sort of expect that maybe even if we looked in biomodels, we might be able to find something. Again, there are people here like Merv Yoder who at the med school who are big experts in hematopoietic differentiation. It's a fascinating problem, and and this question about how do you do validation against large scale knock-in knockout experiments is also quite interesting. I may have mentioned before, this past summer I was at a workshop in Paris, and there was a group that was doing very similar kinds of modeling, and they had a really beautiful framework for trying to do that kind of validation against very large scale perturbation experiments. I think that could definitely be worked up into a very interesting project. With all of these, my only concern is I don't want people to be too ambitious, in the sense that I think you should by all means do things that you want to do. I don't want you to not do the things you want to do, but I also don't want people to get into a situation where it's just too much work. And so if I sound like I'm holding you back, it's not for lack of enthusiasm about the ideas. It's just to try to help make sure that people don't wind up at the end of the semester saying, my goodness, I have to spend 300 hours for getting this model to work. So and I could be wrong. Sometimes I think it's harder than it is, and sometimes there are things where I think it'll be easy and it isn't. So it's not that I always know, but my goal is for people to learn and enjoy themselves as well. So but I think these are all great projects so far, great project ideas.
I intend to research innate immunity as part of my PhD. So naturally, this is the topic I chose. And innate immunity is basically everything available in the tissue to defend against infection that does not have a memory. So if you're infected by dirt, I don't know, this is probably not an infection, but if some dirt gets in your skin, it is able to defend itself against that dirt. But when you get a few weeks, months later, if you get the same dirt on the same spot, it's not like more effective against the dirt because innate immunity doesn't have memory. On the other hand, the adaptive immunity, it has a memory. So it is in fact an intelligent system. If you infect once, it will respond in a certain way. If you infect again with the same pathogen, it will respond better, right? Eventually, the natural killer T cell, which is a specialized immune cell against a specific pathogen, it remains available in the tissue, so you can have it as part of your innate response later on. But you have to have educated your adaptive immunity system beforehand, right? So among the things that are part of the innate immunity are all the immune cells listed here: macrophage, natural killer, dendritic cell, neutrophil, and also interferons, chemokines, cytokines produced by the cell that act both minimizing the virus replication inside the cell, and also signaling to neighboring cells as a way to prepare them to defend against the infection. So the first interesting thing I want to show is the source here. You can find source of networks. You can find networks for your own project. So let me show you. Oh, okay. So I have to request again. Sorry. So this is innate immunity in this site is one of the many other resources they provide in terms of networks. So you will find things about cancer, you will find things about differentiation. So I highly recommend this source, and it is updated and very complete. So in specifically about innate immunity, everything was dated 2023, which is great. And they are very complete, in my opinion. So they are more complete than the papers I read about it. And so I think they are complete. I don't know. I'm ignorant about it, so I cannot judge. And they are very easy to read. So the way the information is presented is very clear, right? So inflammasome, sorry? Yes, unfortunately. So inflammasome signaling, I have no idea what it is. Sting signaling, no idea too, but I heard sting before in some of the other papers. NF-κB, this is a gene regulator, transcription factor, sorry, that promotes, that makes the cell defend itself against viral replication inside it. And I have no idea what this is as well. So I really have to give them a better look. So to prepare myself, I grab a review, and this review is one of the latest I could find, and very complete as well, specifically about influenza infection in lung tissue, and talking about the cytokines, which is my which I'm looking for here. So how the cytokine affect the cell and how the cell respond to the infection, producing cytokines and signaling to and from neighboring cells. So basically, it reviews innate immune response and the role of cytokines, and it gives an extra focus on cytokine storms, which is a major issue in infections because it drives tissue damage. So cytokines, although they are needed to defend against an infection, they promote tissue damage. So they point to ODE models of cytokines, which is of main interest for me. So I will certainly use this reference to find ODE models. And I will probably use the information provided in this reference to build my own models. And then use Tellurium probably to model the pathways and maybe apply it to spatial tissues or compartmental pharmaco-kinetic models. So I highlighted the main topics, for me, the main topics, and I think for you too. So the most interesting things on this review. So the cytokines can recruit immune cells to the infection site. What this means? The infection begins, the cells being infected start releasing cytokines, and the cytokines start recruiting immune cells to the site, to the infection site. Cytokines and chemokines. So they make the immune cells migrate towards the infection site. And the immune cells also release more cytokines. So you have a positive feedback. The more cytokines you have, more immune cells you have, and the more immune cells you have, the more cytokines you have. And this creates this cytokine storm that drives tissue damage. Also, another very interesting information is that the cells have receptors that are activated by PAMPs. PAMPs are pathogen something, I don't remember exactly, but they are molecules specific from the pathogen. So the cells have receptors specifically for these types of molecules from the pathogens, and they induce transcription factors that make the cell produce cytokines, interferons. And these interferons, they make what the name says, they interfere in the viral replication. So they are a major source against early infection in our body. And the same genes and transcription factors that induce the production of pro-inflammatory cytokines are the same that triggers another transcription factors that produce anti-inflammatory cytokines. So in some sense, they do the two stuff. They amplify the infection, but they also reduce the infection. And the balance at which they do both determines the outcome of the disease. So if you only produce pro-inflammatory cytokines, you get a lot of damage. And if you don't have enough, the infection will spread, and the other way around. Then I found specific papers that built OD models. This first one, they model regulation by cytokines and their expression, and they predict cytokine levels in time, and they fitted the results to experiments. So the parameters they used as input to the networks, they are either estimated from the literature, so they are not like precise values, or they fitted using the model and the experimental results. And they also don't consider cell-cell interaction and immune cell recruitment, so that would be a possibility to improvement. This other model addresses mainly cell population, so this also treats the system, the systemic immunity, the adaptive immunity, but it mainly focuses on cell population, so population of T cells, B cells, etc. So I didn't see many of the cytokines involved. And this one is interesting too. It involves either cell population, so the population of infected cells, susceptible cells, and immune cells, but also they also model the interferon one, which is one of the major defenses from the cells, right? So this is more like it, this one, because it either models the immune cells. In this case, they model the natural killer, and they also model as a cytokine. So this is more like it, in my opinion, but is extremely simple. So there are a lot of room here to improvement. For example, the other cytokines, the genes, the transcription factors, etc. And this one, which I didn't understand, so I won't talk about it.
Okay, well, that's a lot of different possibilities to look at. Any one of those by itself is already a big project. I personally think it would be very interesting to see the model of the intracellular signaling. And I would say the context of one semester in the class like this, I wouldn't try too hard to integrate the subcellular signaling with the population dynamics. But there's nothing wrong with that. As you point out, they're very different styles of model. They're very different philosophies of modeling. And the Perlson paper that you cited is quite famous. Alan likes very simple models. So you could tell. You might not think mathematics has a style, but it does. Just the way. It's not that you could always tell who's done what, but often there definitely are house styles, and that Alan has a particular house style. He's of course one of the great modelers of infectious diseases. But I think trying to understand the toll-like receptors and the intracellular signaling could be quite interesting. As you point out, when you look at the actual networks, like the NF-kappa B response pathways are pretty intimidating. The inflammasome response, there was work done by Mark Chaplin's students at Dundee who worked with Paul on that, and Michael Getz, who just moved into our group from Paul's group, has worked on this as well. So you might want to talk to him as well. But again, I think for everyone, I'm not trying to discourage you. I think it's wonderful that you're thinking in very ambitious ways, and I want everyone to learn. But again, don't feel that for the purpose of this class for this semester you have to do something enormous. Something modest and achievable is just fine. And if it happens to be something you can then extend later on for your PhD or for other research, that's wonderful. But the main point is to learn. So they're great. Those are all great problems. Bart Ehrmantraut has published some nice, very simple models of. In a sense, what we're talking about with infection is not so different from what we were talking about with cell cycle, which is early in infection, you need to have rapid upregulation of immune response, and then you have to have some way of turning it off. And so typically what you have is that those early excitatory positive feedback cytokines and chemokines act over short time scales, and then the inhibitory anti-inflammatory molecules turn on more slowly but are stronger in their effect. And so essentially at the beginning the infection starts, you turn on both pro-inflammatory and anti-inflammatory signals from the beginning, but the anti-inflammatory signals take a long time to build up. And so at a certain point, the pro-inflammatory signals come up, the anti-inflammatory signals begin to come up, and then they get high enough, and then they turn everything off. And that doesn't always work, which is how you wind up with septic shock, if it's a cytokine storm, or chronic inflammation, which is a different pathway. And actually, TJ Sego, who's in my group, published a nice paper with Bart Ehrmantraut and a group of undergrads who actually did it as part of a class project at a class like this with Bart. That was a paper they published at the end of the semester, modeling how you could get either immunosuppression in which the virus takes over, sort of an ideal immune response where you get the virus comes up, the immune response comes in, it gets rid of the virus, and then turns off. Cytokine storm, where the immune system takes over and kills you. And then also chronic inflammation. The chronic inflammation one is usually the hard one to get. It's relatively easy to get. The virus blows up, the immune system blows up, or everything works. It's a little harder to come up with a system where you could wind up with continual inflammation that doesn't blow up. So
yeah, it seems a difficult equilibrium
right
to achieve.
but the But Bard's models are very abstract. He tends to ask the question mathematically, what do you need to do to get that kind of response? And then after he's done the mathematics, he says it must look like this, and then you look at the molecules to see what could be doing it.
Yeah, but that's very interesting.
And so Bard has incredible insight into the mathematics of these things. But here in Indianapolis, Julia Arciero has published papers of that kind. She worked with Alan Perlson. So she has so there definitely are people around here who have that kind of insight.
So I'm particularly interested in gene regulatory networks, and I find this paper for gene regulatory networks for myeloid progenitor differentiation. And first, I have a huge love for Boolean gene regulatory network, and that goes back years. I think I read really years. Before 2000, I read about them before I even did bioinformatics. I think they're really very interesting how they work and what they do. And this is the book from the person who really was, I don't know if he invented them, at least for biology, or drove this whole way of thinking about Boolean gene regulatory networks, and that quite a success to describe gene regulation with these networks. And so this is one of my scientists that I very, very admire. I read this book. I didn't understand half of it because it was there too mathematically for me. I was just working as a technician for years, and now I have the chance to do my PhD. I'm now the second year PhD student, and I learned the math, and I understand more and more what really goes on. So in this class, I work with agent-based model normally. I work in Paul MacLean's group, but in this class, I have now really the chance to do modeling in this direction. That's why I really, even with Tellurium, would like to go towards gene regulatory networks. So as said, I'm in Paul MacLean's group, and the PhysiCell is the software that we work on. And luckily for me, there is a plugin that's called Species, and it's derived from Mabos, and these are collaborated with our groups. So we have a plugin to do gene regulatory network analysis. And this is just from the background that I want to learn this technique more, because at one point I really would like to investigate those models in my research. So, but now about the paper. So I found this paper, here are the differentiation of myeloid cells are encoded in transcription factors. And it's an interesting model because it's not too big to implement. This is the network. So there are 11 genes, there are 28 links. And this model this is from the Thais lab. So the Thais lab is in German lab, but they wrote quite a nice Python soft analysis package for single cell data analysis. And they have among others, they have deeply analyzed this model. So the reason that I would like to choose this model is, can I take this model, implement it in Tellurium, and reproduce what they have done and understand their findings? And then I would take it's a gene regulatory network, so we have kind of like step functions. So then we would relax the step function and then go more towards a more gene regulated, but not a Boolean gene regulated, more like with different expression level regulated model, and see how this will change our results. And then one other thing that we could add is that we have what is called an stochastic Boolean gene regulator network. So when you not anymore every time really switch, but you have a certain probability that you really switch your gene or not, because Boolean networks have some restrictions. The basic network has just a time clocking that is very given, and this is not really how the nature works. The nature doesn't work so clockwise. So my big interest is really to work with this model and understand Boolean regulatory networks, understand as well networks in general, understand the when I have a relaxed function or when I have a step function. Try out all these things that I read in the years before now by myself. I'm quite pragmatic with this class, I have to be honest. Yeah.
Go ahead, Justin.
With Boolean networks, are they similar to Markov models or hidden Markov models chains?
So
Go
should I? So a Markov model is
Please repeat the question so that whoever's
Okay.
online can hear it too.
So Justin asked about what is the difference from a Markov model to a Boolean regulatory network. And so, okay, we talk about Markov model, and Markov model are reaching a steady state, and they have the probability from one node to another node, and they will reach a steady state. So on the Boolean chain regulatory network is a little bit different. So what you have is you have Boolean gates like and or. Do you know? Are you aware about that? No. Okay. Do you had set theory once in primary school? Also set, so like when you have a intersection or when you have a union. So a union more or less is an OR. So it means when you have one gene and you have as well the other gene. So then both of them, either you have one gene that is already says the gene is you have to think about the minimum is 3 genes. So you have one input gene, another input gene, and an output gene. And when you have a union from the two input genes, then it means if either gene A is on or gene B is on or both are on, then the output is on. Now you can as well have an AND, which is the intersection. So you can say gene A is on, but gene B is not on. Then there's no intersection, so then the output is not on. But when gene A and gene B is on, then the output is on. So these are just the two basic Boolean functions. Then there is a Boolean function not. That means just when you have on, then it's off afterwards. And when you do the maths, you can have 16 different basic functions. And of course, sometimes you have not only 2 genes as input, sometimes you have more as 2 genes as input. But from these basic Boolean functions, you can drive the whole network. So it's not how computers work. They work a little bit different, but it goes into direction how at least an automaton works, which is the basic way how computation is done. And it's more like a Turing model. The modern computers are a little bit different. The von Neumann machines is a bit far away. It's much more primitive. It's much more like chain regulatory network is done, and it's just very, very interesting to be able to think about that and study the biology. That's where I would like to go into it. Yeah.
That's great, Elmer. It's interesting that 2 of you picked the same core
Yes, that was
toggle.
fun.
One of the classic differentiation inducing toggles. We tend to do the SOX-OCT toggle in this class, but that they all function very similarly. But that core is actually a good example of this in the sense that, right, you either have GATA1 turned on or you have P1 turned on, right? That you're unlikely to have both, although I suppose it's possible you could get an intermediate expression of level of both if they're being driven in a particular way. And so the fact that you have one or the other is why a Boolean approach tends to work. And yeah, you can certainly use Markovian methods on that as well. And so, I don't know,
I
maybe
s-
the two of you should talk some more about this, because these differentiation pathways have a lot, they're never the same. Every tissue is slightly different, but they have a lot of overlap.
I think it will become very interesting if you go different roads. It will be very interesting to compare. And if we work together, then we exchange it there. So both is possible.
I think that's great. I mean, they're all great projects. I have to say I wasn't expecting such elaborate presentations today. I thought people would just show this is the paper and say a word or two. Hayden was saying in the chat, we don't really need you to do another presentation next week. So perhaps instead of that, we could have people develop these a little bit more, maybe meet with me one-on-one a little bit, and we could talk about them. I mean, again, I certainly don't want to force people to work in teams. In my experience, when you can team up, the downside of a team, of course, is that unless you're lucky, there isn't somebody who else wants to do exactly what you want to do. On the other hand, especially in a class like this, where we have somebody who's two people who are very biological, some people who are more computational, some people who are more software. Sometimes if you have a team, you can complement each other. But it's also fine to talk to each other without having it doesn't have to be a formal collaboration. It could also just be, since we're a small class, we could work together on these things. So just to go over quickly, review, last time we talked about a lot, actually, covered a lot of material. We talked a little bit about PBPK models and what they can do. We didn't really go into the mathematics of PBPK models. I hope maybe later in the semester we'll have a chance to talk about how they work mathematically. We introduced some basic chemistry and the concept of stoichiometric amounts. And there in the homework were some examples of using stoichiometry. And it can be a little bit confusing if you're not familiar with these before. You have to get some practice. If you say have two molecules of ADP making ATP plus AMP at some rate VF, you have to know how to use that too. And you can have reversible reactions, and you have to know what to do when you see a reversible reaction as well. And there is a straightforward, although not always easy to remember, set of mechanical rules that you have to do. The first thing to do is to remember that the left-hand side of your chemical reaction is the positive side, and the right-hand side is negative. So going from right to left, you lose on the left and you gain on the right. And then the reaction rate is normalized by the stoichiometric coefficient, so that the same rate applies to all the rate applies to the reaction, not into the individual components. And so if you want to get the rate law for the individual components, you have to multiply by the stoichiometric coefficients. And the homework that was due last week was done before we had discussed this in detail. And so I know some people found that a little bit confusing. And so that's why I wanted people to practice a little bit, just to develop familiarity with that. So if you have a reversible elementary reaction with A molecules of A plus B of B going to P of P plus Q of Q, well, if each component appears only on one side of the reaction, then the A by dt is equal to A, the stoichiometric coefficient, times the forward rate, the backward rate minus the forward rate. And you do that systematically. If the component appears on both sides, then you have to multiply that V reverse minus V forward by the number that you destroy, which is A on the left-hand side, minus the number you create, which is the A on the right-hand side. And so in principle, there are 4 components for a reversible reaction that has the same species on both sides. You have to rearrange 4 numbers to make it work. And I refer you back to this slide because once you've seen it, no matter how complicated it is, you just say, take this one, put it here, take this one, put it here, take this one, put it here, and so on to rearrange it. Okay. Then we also talked about mass action rate laws, and we'll come to other rate laws. I hope, maybe given where we are in terms of time, maybe not today, but the plan was to come today. And the basic concept of mass action is simply that for chemicals to react, for molecules to react, they have to get close to each other. The probability of a molecule being in a given volume is proportional to its concentration. If you need 2 of them, then that means that the probability of 2 of them being in a volume is the concentration squared. If you need 3 of them to react, the probability of 3 of them being at the same place at the same time is the concentration cubed. Now that's assuming that there's no excluded volume, so that the concentrations are fairly low. The molecules aren't too big. But that's the basic idea. If I ask what's the probability that there's 1 person in this room, if there's a density of people in the building as a whole, well, that's the room volume times the density of the people in the whole building. If I want to know what the probability of 2 people being in here is, then that is the probability times the probability, as if the probabilities are small. If the probabilities are bigger, then I have to worry about the sort of the complicated cross terms. And so that, but so mass action assumes I can ignore those cross terms. If I have a irreversible reaction, I just write the rate only depends on what's on the left. Similarly, the back reaction depends only on what's the right. And so the net mass action is the rate, forward rate minus the backward rate. And so you do them separately. Basically, you don't ever think about a reversible reaction as both things together. You analyze it as the 2 parts separately. So you say, this is an irreversible forward reaction and then an irreversible backward reaction, and then you add them up. And then we also talked about boundary species, and boundary species, it seems like making a lot of fuss really over nothing. A boundary species simply is a species which you assert doesn't change in time. So if I have a source of a chemical and I'm adding chemical at a constant rate, then that would be a boundary species. If I say I have a big reservoir of a chemical so that the amount of that chemical doesn't change when I do whatever my reaction is, then that's another example. I mean, in this room, the volume of the room is big enough that the amount of oxygen I'm consuming doesn't really change the amount of oxygen in the room very much. Of course, in reality, there's always some change, but I can neglect that to first order. And so, in particular, very often we'll write things in terms of having the nothing go to a chemical species. We won't ask where it comes from. And very often that rate will be a constant rate. We'll have a constant supply. For decay, we talked about this. So this came up a little bit in the homework because we hadn't really gone over it. Decay almost always is multiplied by the concentration. And the reason for that is if you had 0th order decay, if S went to nothing at a rate K, then if you had a small amount of S would go negative because you'd use it all up and it would keep reacting. And so typically decay reactions are multiplied by concentration. Now, I mentioned last time, and it was in the homework a little bit, maybe we belabored it a bit much, this is more for people who have to read these things in journal articles than anything else, that there tends to be a amalgamation of these chemical reactions, like the one that I've described here. I have 3 molecules of A reacting reversibly to make 2 molecules of B. I also have a source that's creating A and a decay of B. And the way it's written, I have nothing producing 3A at a rate K1 and 2B going to nothing at a rate K2B. And one has to know that when it's written that way, you don't multiply the K1 by 3 or K2B by 2. You really, that's an abbreviation with nothing goes to A, 3A goes to B, 3A goes to 2B reversibly, and then B goes to nothing. And so that's a shorthand that's a little bit unfortunate because it's inconsistent. It's mathematically inconsistent, but people use it. And so I'm just reminding you that that exists. And so that example here, when I split them up, I can write the chemical reactions immediately. The key thing to notice here is what I just said, which is this number 3 for A shows up that as the rate is KF A cubed. Because there's a 3 in front of the A, I lose 3 molecules of A. So that 3 from in front of the A comes down as a 3 here in the rate law for A. I'm creating 2 molecules of B, and so that 2 comes down into the rate law for B as 2 in front. And so there are not that many things I have to remember, and it's consistent, but until you've practiced it a little bit, it can be confusing. William, go ahead.
Yeah, wait, I'm confused. Why is it 3KRB squared? Shouldn't it be 2? Do I not understand?
That's a good question for A. So that's the thing that also is a bit confusing. The first equation, the first rate law for A is the concentration of A. If I destroy B, I create 3 molecules of A. And so the number that's in front here for 3 and 3 is the 3 from the A here. And so I can see that you might expect it would be 3 in the first one and 2 in the second one. But if I think about, suppose I go forward and backward, I create 2 molecules of B and then I destroy 2 molecules of B. My number of A shouldn't change. And so if I want to be able to go back and forth and keep my mass conserved, I have to do it that way. But that's why I gave you those practice examples where it was a question of writing it out, because I agree it's really easy to say, oops, to write the 3 in the wrong place or the 2 in the wrong place, especially if you have A on both sides of the equation. Suppose I'm destroying 3A and creating 2A. Then I have to remember who gets multiplied by what. So that's a very good question. In general, we're not going to do a lot of complicated stoichiometry in this class, but there are plenty of times when you do. You'll have some 4 components of hemoglobin come together to make the complex, something like that. So it does happen. Any other questions about that? Because that's a pretty fundamental point. I think I want to make sure that people are comfortable with that. Again, I know looking at the homeworks that people submitted, people found that a little bit confusing, and feel free to, you don't have to get perfect scores on all the homeworks, but it is important to be able to solve the homework problems. And so if people have something on the homework where they want to go back and do it again and submit it as another time, I'm happy to give people to regrade it. But again, the main point of the homework, I mean, they are a part of your grade, but the main point of the homework is to help you learn. So, and there are additional exercises in Herbert's textbook one could do as well. Now we haven't talked much at all. We haven't really talked much at all about coding. Last week we wound up talking about biology all week, which was nice. I happen to love biology, so I don't mind it. But it is a course in modeling and simulation. So there were a couple of basics that we talked about with Tellurium last week. In particular, how do you read and write antimony values from Python? And the answer is if you loaded a model in, you use whatever the model reference or the model handle is. So if I loaded the model under the name R dot A would be a reference to the variable antimony variable named A. And I can read from that or write to it. It doesn't have to worry. Simulate has the feature that it leaves the system in a state which you can continue from. So if I call r.simulate 0, 10 once, it'll start from whatever the initial values were in the antimony model specification. If I then call r.simulate 0, 10 a second time, it will start from wherever the model finished off with the values that were the final values and continue. So this allows me to run the simulation, see what happened, and continue it. But that means that if I want to start over, I have to use a reset function to tell Tellurium to put the values back to the ones that were in the original specification. So by default, time continues. You have to reset it if you want. And then for display purposes, we talked about this plot array, which is basically matplotlib, a shorthand for some matplotlib functions. And there were a couple of functions that we were going to use quite a bit for convenience. One was show equals false or true. Show equals false tells Python that we want to overlay our plots, put everything on the same plot. Show equals true will actually display the plot on the screen. And then there was this not very elegant reset color cycle equals false, which says to use a different color for each plot. And then the way of adding labels to the plot, where you add labels and then a string that's the argument of the labels function. And that'll give you a key. It's possible to move where that little key for the thing displays, put it inside or outside. There are a whole variety of options, which you can look up in the manuals. If you want to be fancy, you can be fancy, but for the purpose of class, I wouldn't worry too much about the fancies. If the labels are overwriting the thing you care about on your display, then you have to learn how to move them around so they're not your way. So I'd like to, and then the last thing we did, we ran over this a little bit quickly last time, was the difference, concept of a steady state versus an equilibrium. And so let's start out with a system like A goes to B and B goes back to A. In this case, there's no mass being added or taken out. So the total amount of material that I have is going to be A0 plus B0, whatever I started with. And if I wait a reasonable amount of time, I'm going to get the final or equilibrium value B divided by equilibrium value of A is some constant. And that the bigger that constant is, the more B I have and the less A I have. And it makes sense if I just look at what's happening. If I have KF be larger, I'm going to have less A. My forward rate is higher, so it means I'm destroying A faster. If I have KR bigger, then I'm destroying B faster. And so maybe it's not a surprise that B over A is going to be KF over KR. And that has a name that has the name of an equilibrium constant. What may be a little bit more surprising is that if I have a very complicated reaction, A plus B plus A molecules of A plus B molecules of B goes to P molecules of P plus Q molecules of Q, dot, 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 dot. And the equilibrium is still the same. The K equilibrium is P to the P times Q to the Q times et cetera, on either side, and it's still just KF over KR. So this whole complex thing, from the point of view of equilibrium, behaves as if there's just a single component on either side. And I'm not going to do, I'm going to maybe do a little bit of mathematics this class, more than I normally do. But if you want to see the derivation of that, you can look it up. I'm not going to take you through it in class. But in the math bio textbook, if you're interested, you can see how to do that. One thing that we'll run into just notationally, and I understand that telling you about notation like arrow inconsistencies or things can seem rather dry, but since you're going to be reading papers written by a lot of different people, I want to give you a heads up about some of the things that may be confusing when you're reading those papers. And so when I have these comments like people use arrows inconsistently or label them inconsistently, the reason they're there is because in the past I've had students who got confused trying to read a paper, or I got confused trying to read a paper. And so one of the things that we typically use the standard capital letter to represent the molecular species and the italic capital to represent, like capital A is this molecule A, italic capital A is the concentration of A. Well, the problem is that in chemistry, we tend to write the complex of a pair of molecules by writing their names next to each other. And that gives us a problem if we are writing the concentrations, because the concentration of the complex ES should be italics ES. But of course, multiplication is also written by putting the things next to each other. We don't always write a dot or a cross or something else. And so there's an ambiguity here. So I'll try to always write a dot. E dot S means concentration of E times the concentration of S. ES by itself means the concentration of ES. Okay. Some people like to put the concentrations in square brackets to distinguish the species from the concentration of the amount. And the brackets have the advantage that they make the difference between E times S and ES clear. The disadvantage is that they tend to accumulate. So you get lots of things get pretty long when you have lots of brackets. But you'll certainly see that as well. And occasionally people will use lowercase italics for the concentration, but the uppercase is typically for the species. Again, Herbert's textbook tends to be pretty consistent about these things. Early editions were not. He's worked hard to try to make it consistent, but in the literature, it's not always consistent. Let's come back to this reaction, and we started out with A going to B and B going to A, and that will equilibrate. Now I'm going to have a source of A, so I have nothing going to A, and that'll be, let's go to the 0th order source. I'm pouring A in at a rate K1, and B is decaying at a rate K2 times B. Now, if I run this situation 2, I'll reach a situation in which the concentrations of A and the concentrations of B don't change, but it's not a steady state. It's a steady state, but it's not an equilibrium. There's no chemical equilibrium. I'm always having a source and I'm always getting rid of the chemical. And so there is no equilibration here. And right, there's the brutal observation that you only have chemical equilibrium when you're dead. And even then, it takes an awfully long time, because actually decay takes a long time. I remember in the physics department, when I was in the physics department, somebody left a pot of coffee sitting in the seminar room for years. I mean, really for years. And they sealed it, they covered it. And at first there was this nice layer of mold that grew on it. And then that layer of mold collapsed, and then some other kind of mold grew on it. And then there was something that looked rather bacterial growing in it. And over a period of about 8 years, there was a succession of different species of various things growing in the coffee. And then eventually it was just sludge. I think every possible nutrient that they could have had in that coffee and the products of the various organisms living in it had been used up, and nothing could live in it anymore. At that point, somebody took the pot of coffee and dumped it out.
The creepier story is when nothing of this happens.
That's
Usually.
and then somebody washed it out and they started making coffee with it again. But in any case, that was it. But it took quite a while. That's all I'm saying. But it was sort of funny to. It started out that somebody simply forgot to clean the coffee pot, but it became sort of an item of interest to the department to watch it. And there actually was Stan Leibler at Rockefeller University did a whole series of really interesting experiments where they would seal nutrients and various unicellular organisms, bacteria usually, into a container and then watch what happened. And it's actually remarkable how difficult it was for, you'd think you put, say, glucose and water and a little bit of oxygen in a container, and then you put E. coli in with it and you watch what happens. And you think that would be relatively repeatable. Well, oxygen diffuses through glass. CO2 diffuses through glass. The amounts of the nutrient weren't exactly the same. The temperatures were not exactly the same. Light had an effect. It was amazing how many different trajectories just that same, apparently identical experiments could have. Really, really an interesting set of studies. Quite beautiful. That was the Leibler lab was sort of specialized in those kind of apparently very simple, but in fact quite tricky experiments. Mike Lynch, who left IU for Arizona State University, did a lot of beautiful experiments of that kind here. And there are groups here still studying bacterial evolution, for example, that do this kind of work. That's getting us pretty far from the simple mathematics of a chemical reaction, but it's not unrelated to the things that we're going to be dealing with. So mathematically, there are a variety of terms for these things. Steady state's also known as a fixed point. And simply, if we have a rule that dA by dt is equal to some function of the concentrations, then any time that rate of change of A is 0, we have what's called a fixed point. And if we have a set of concentrations for which all of our net reaction rates are 0, then the concentration won't change. And potentially there could be many of those. And of course, in more complex systems, we can have situations which the cell could be in one of multiple stable states. In a sense, that's what we mean about when we're talking about differentiation. And so the study of what the possible steady states or fixed points are for a given set of parameters is rather elaborate. That's really a big part of what dynamical systems, the mathematics of dynamical systems is about. Yeah, William.
How can there be multiple fixed points if a fixed point is one, or is it? Oh, I guess just in a graph.
Well, let me take this notebook.
Yeah.
If I drop this notebook here, it's going to fall on the floor.
Yeah.
If I put it on this table, it's not moving. If I put it on the floor, it's not moving. Gravity is acting on it in either place, so the force on it is the same. The mass of this is the same, but the system has two places that can catch it. There also, and later in the class we'll talk about this, Herbert has a pretty good chapter on it, and some of the other textbooks go into more detail about the difference between a stable and an unstable fixed point. So the table is an unstable fixed point. If I push this a little bit and this falls off, it will fall to the floor. The floor is a stable fixed point. If I push it on the floor, it doesn't come up. And so we can analyze whether a given fixed points are stable or unstable. A key thing is that as we change the parameter values in our models, fixed points will appear and disappear. And when that happens, that's called a bifurcation. And that typically is pretty important. For example, if a cell is in quiescence and I push the cell into cell cycle, that would be a change of, I mean, cycling isn't really a fixed point, but you can describe cycles as something that's periodic and repeats. Can be thought of as a kind of related to a fixed point, if periodic motion is related to fixed points. And so you can have transitions from a fixed point to an oscillatory state, or from an oscillatory state to a chaotic state. Those transitions are also bifurcations as they change parameters.
Yeah, but isn't that with a third party, like the third party action is making you can't, you know, do you understand what I'm trying to
So Williams says, well, but who's changing the parameters?
Yeah.
Which is a good point. So typically in dynamical systems, yes, you assume that the parameters have fixed values, and then you ask what are the steady states or oscillatory states or other states for those values. And then by hand you change the parameter value. But you can imagine, for example, the cells in your body are making the choices they're making, and those are autonomous. Those are inside the cells. You're also getting older. That's changing parameters in your body, and slowly, in a way that's not the responsibility of the individual cells, or maybe actually be a reaction, may be partly contributed to by the cells, but it's a little bit outside of the model of the cell itself.
Okay.
But that's a very good point, that what's a parameter and what's a variable depends on your point of view.
Well, yeah, no, I was just thinking about it in terms of like the reaction that was just on the previous slides.
Right. Right. Well, the reaction on the previous slide doesn't really have any exciting. It only has one fixed point. If you don't have feedback, you're not going to get anything exciting. Those networks that we were seeing about differentiation are classic examples where you can have molecule 1 high and molecule 2 low, or molecule 1 low and 2 high. And those are two states of the same cell. So you can choose between them. The classic in physics, those tend to be called phase transitions. And an example of that might be a traffic jams, are a classic example of that. But phase transitions like water to ice to steam would be examples as well. There you're changing the temperature. The temperature is an external parameter. And the pressure. All right. So let's just quickly review this homework problem, where I said, what are the steady states of this chemical reaction? And I don't want to drag us through too much of this, but I do want to at least go over one or two of these just so people have a sense of how it goes. and we can take a minute to think it through. So the first thing we do is we look at that reaction diagram, and we have to write down what the rates of change of A and B are. And I just start on the left. There's no stoichiometry here, so I don't have to worry about weird interpretations of the chemicals of the arrows. I have nothing going to A, so that means here, down here, I have a source, K1. dA by dt is equal to K1. A is turning into B at a rate Kf times A. That means I'm destroying A at a rate Kf times A. B is turning back into A at a rate Kr times B, so I add Kr times B. So I have three arrows that affect A. The source of A turning into B, and B turning into A. So I have three terms in my ODE. Okay. Now again, if I had stoichiometry, I had 3A and 6B, that I'd have some extra numbers floating around, but here I don't. For B, let's walk through this. I have three arrows pointing at B. dB by dt, well, every time I destroy an A, I create a B, so I have Kf times A from the top. I'm destroying B with the back reaction, Kr times B is the second one, minus Kr times B, and B is being destroyed at a rate K2 times B, that's a decay. Now, how do I find the steady states? Steady states are found by setting dA by dt to 0 and dB by dt to 0. So I'm going to write these two equations on top of each other with zeros on the left-hand side. And you'll notice what's called the balance, which is there's Kf time, and sorry, another notational thing. The steady state values are often written with a star. So A is the changing value, script A, italic A is the changing value of A star is the steady state value. So the star means that I'm looking for the steady state, okay? Stars are used to mean a lot of different things, but that's one of their uses. And so now, is it clear how I go from the ODEs to this pair of equations here? Okay. Now, what's called the balance is, you'll see Kf A star here, and the next line Kf A star. This is the destruction of A with that forward arrow. This is the creation of B from the same arrow. Kr times B star, Kr times B star. So you'll see pairs. Every arrow will create a pair of terms, which would look the same. Now, if there were stoichiometry, one would have a 3 in front of it and one would have a 2. So it might not be so obvious here. But here they're pretty obvious. And you'll notice that most of these terms are going to cancel. So if I add these two equations term by term, those terms cancel out. And I'm left with 0 is equal to K1 minus K2 times B star. Does that make sense? And that means I can solve for B star. Does it? That second equation is algebra that's simple enough that even I could do it. K1 minus K2 times B star is equal to K1 over K2. Now I realize that when you have, if the rates are not linear, suppose the rate was Kf times A cubed, or 7A cubed, then the algebra you have to do to solve it might be more complicated. But conceptually, the finding steady states isn't any more difficult than what we're doing here. If it's a very complex equation, you may have a mess, algebraic mess, but the concept is what I want to get at, which is if we're looking for a steady state, what we're going to do is we'll write our equations and then we'll look to sum them up, because all of those A goes to B, B goes to A will cancel out. Notice the only thing that's left here that matters is the source, K1, and the destruction at the far end. So those intermediate A goes to B doesn't do anything. It's disappeared. Which is sort of interesting. You might think that the amount of B would depend on the ratio of KF to KR. But you'll notice there's no KF or KR in the amount of B. It only depends on the source and the destruction. That's actually a pretty fundamental observation in chemistry. So if I have some long set of chemical transformations, I have a source of something here, it gets destroyed over there, and I have some long set of transformations in between, and I want to know how much at steady state do I have. The answer is none of those intermediate reactions make any difference. It's only the amount that I put in and the amount that I get out. Question, William.
So equilibrium would be without the source and sink. It would just be, and
Right.
that system would be in equilibrium.
So William says, what if there were no source and no sink? The answer is if there's no source or no sink, to calculate B star and A star requires a little bit more work because it will depend on the initial amounts of A and B. The other thing you'll notice here is that B star does not depend on how much A or B I started with. Basically, the system forgets because I'm destroying, because of that K2B decay, the system forgets how much A and B I started with. The final amount does not depend on how much I started with. I think that's not something that's immediately natural to people. I think it's worth thinking about that a little bit. Basically, the system flushes out its initial state. The final state is independent of the initial state. So that was a good question. Okay. And then the last thing is I can have to solve for A. So now that I've solved for B star, I can plug it back into my equation for A. And then it winds up that the equation for A is a little bit more complicated. That intermediate one, I don't think I want to walk us through all of that. There's a little bit of algebra to solve for A. And you'll get something that looks like this. And you'll notice here that the amount of B depends only on K1 and K2, the source and the sink. The amount of A does depend on the other constants, because basically, if you think about it this way, A is sort of is an intermediate. If KF is very big, I don't stay A very long. I pour stuff A in and I get rid of it right away. And so if KF is large, I don't wind up with much A. If KR is big, then the time it takes to go from A to B and then disappear is longer. And so if KR is big, I wind up with more A. And so one can play around with reorganizing these a little bit to make them clearer. If the final form that I think is easiest to look at, depending on what you're going to do with it, you could arrange it a little bit differently, is this one, where here A star is equal to K1 over K2, that is B star, multiplied by KR plus K2 divided by KF. Well, one way to think about these things, if you don't want to have to do the algebra, is ask the question, if I increase a parameter, should it increase or decrease the amount of A? If it increases the amount of A, then it's going to come in the numerator here. If it's going to decrease the amount of A, it'll go in the denominator. And so here, if I increase the amount of the K1 and K2 are coming outside, but what about KRF? If KF is big, I'm destroying A fast, so I'll get less. So KF is in the denominator. KR is creating more A. So if I increase KR, I get more A, so it goes in the numerator. The one that's not so obvious is why would having more K2, why would destroying B give me more A? That's the one that you probably couldn't guess looking at the diagram by itself. That's the one where you probably have to actually do the algebra. But at least the KR and the KF are probably, you should be able to figure it. And if I want to know how much, how much A and B I wind up with, I can add those two things together. I have K1 over K2, so I'll get K1 over K2 times 1 plus K da 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 da. And if I multiply that out, maybe I'm doing too much algebra in a slide. But I wind up with this, which is K1 over K2 times KF plus KR plus K2 divided by KF. And the whole point of this was actually to compare my two cases. So in the first line, I have A going to B reversibly with a source and with a sink. And the second one, I have the equilibrium case. And I get things that look very different. In particular, and I don't want to go through all the math. You could sort of compare them line by line and ask, how are they the same or different? But there are a couple of things that are I think, worth thinking about. One of which is the driven steady state, that's the first one, doesn't depend on my initial concentrations. The amount of A and B I wind up with is independent of how much I started with. The equilibrium one, the amount I wind up with depends precisely on how much I started with. You notice in the second one, the amount at equilibrium I have depends on A0 and B0, the initial amounts. In the case of the driven system, the first one, the amount of B that I wind up with depends only on the source and the sink. It doesn't depend on KF and KR. In the equilibrated one, everything depends on KF and KR. Can't depend on K1 and K2 because they don't exist. They're not there. The ratio of A star to B star, the ratio of the amounts, for the driven system looks like KR over KF, KR plus K2 over KF. So that doesn't depend on my source, doesn't depend on K1. The ratio for the equilibrium has KF in the denominator and KR in the numerator, but there's this extra term K2. And so there's similarities, but they're not the same. And so when you want to talk about steady state versus equilibrium, there are certain aspects which are similar. Bigger KR means you get more A equilibrium A. Bigger KF means you get more B. That makes sense. But the other dependencies are quite different. Yeah, William.
Well, on that slide, for the K2 in the numerator,
Yeah.
you shouldn't think of it like increasing K2 will increase the amount of A. You think of it like increasing the amount of K2 will decrease the amount of B.
Precisely, exactly. Because you're only looking at the ratio. Very good point. So William says, actually, I explained it wrong in that A star over B star equals to KR plus K2 over KF. The reason K2 is in the numerator is that K2 is reducing the amount of B star. And so it's increasing the ratio. But I shouldn't be thinking of it as increasing the amount of A star. I should be thinking about it as decreasing the amount of B star. Good point. Very good point. Thank you. But that's great. I mean, that's why I understand that sort of seeing a lot of algebra on the screen isn't very exciting. But if you can look at these things and begin to think about things that way, that's really valuable, because then when you come to more complex systems, you'll have an intuition. Does this make sense? And one of the things that I think is quite important and is that when you write computer simulations, well, the simulation could crash because you made a syntax. There's a syntax error. But in general, you fix the syntax errors. And so it will give you some answer. But that answer may not make any sense. If you wrote the wrong rate law, or you typed a plus where you meant a times or something like that, the answer won't make any sense. But it'll run. And so you have to have some way of knowing, developing an intuition, does this answer make sense? And so looking at what these called analytic calculations, for very complex systems, it's usually pretty hard to do these analytics. But you could at least begin to ask the question, if I increase K1, should A2 increase, A star increase or decrease? If I decrease this or turn this to 0, what should happen? And then if your simulation, gee, when I increase K1, A star, I increase K2, A star over B star should increase. I increase K2 and A star over B star doesn't change at all. Well, I know there's something wrong. And so having a sense of what's a reasonable behavior, pattern of behavior, even if you don't know the exact functional form, is it A squared, is it K squared, 2 squared, is it K2 Q square root? All I need to know is that if I increase K2, the ratio A star over B star should go up. That's something I can check and develop a test for. And this is something that biologists do all the time in experiment, because you do differential experiments. You add a chemical. Should the rate of cell replication go up or down? If I knock out a gene, should the rate of cell death go up or down? And so this idea of doing differential measurements, you may not know the exact numbers, but you should at least know, if I change this in a given direction, the output should go up or down. Those are very powerful ways of checking whether your simulations are doing what you expect them to do. And so I think that's a great point for me. Okay, so let's talk about enzymatic reactions. So one of the classic things that happens in biology is an enzyme, and we'll give the enzyme the uncreative letter E for enzyme, and it's going to bind to a molecule which we're going to call substrate S, and it will produce a product P. And the key thing about an enzymatic reaction is that the enzyme is not used up in the reaction. So I start out with E plus S, and I get a complex ES, which are bound together. And then there's a second reaction, a decay reaction, where the E plus S is released to make E plus P. Now, in reality, I could ask this question here: is this stoichiometric? And Elmer says yes. It's a little bit tricky here, because if I really was just to be strict about this, that would mean that S is the same as P. I guess I could rearrange the components of the atoms at S, but in reality, you'd expect P to be multiple species, for example, because otherwise, where does how can S and P be different? I mean, from a chemical point of view, there's nothing, no opportunity for it. So in reality, this is probably not really a stoichiometric reaction, or it's really an oversimplification. I'd actually have S1 plus S2 on the left and P1 plus P2 on the right, something like that. In any case, from the point of view of the mathematics, this is fine. From the point of view of biochemistry, it's a pretty weird thing to write. So if you're coming at this from mathematics, you look at it and say, it makes sense to me. But if you were a chemist and look at this, you'd say there's something screwy about it. Okay. But we're going to assume mass action, which means that the forward rate is K forward, a rate constant times the amount concentration of E times the concentration of S. The backward rate is just Kr times ES. And this is why you have to have that dot, because if I left the dot out, I couldn't tell the top from the bottom. This is why I mentioned this problem about writing complexes. And then the decay is also just a rate constant, decay rate times ES. So S is consumed and produces P. The total amount of enzyme is conserved, which means that E plus ES has to be a constant. And this has a fancy name. This conservation law has a fancy name, which is a conserved moiety. And why does it have this fancy name? Well, because ES has E hiding inside of it, right? It's not, the enzyme is not, ES is not the enzyme, it's a complex. And so I have to know that the complex is composed of the enzyme and something else to be able to know that the enzyme is conserved. And knowing that the total amount of enzyme is conserved, just the way when I did the equilibrium, the total amount of A plus B was always the same in time. I could move material from A to B and back, but I didn't destroy or create material. It was stoichiometric. In this case, the amount of enzyme is conserved, but I have to know that it's hiding in ES. Occasionally, it'll require me to be a little bit careful when I'm doing arithmetic to avoid getting 0 divided by 0. And in numerical solvers, sometimes it can also cause problems. And it used to be that Tellurium required you to specifically say, check to make sure that check for conserved quantities. These days it does it automatically, but some of these slides still have that old syntax. Okay. So now let's do this out. We have how many chemical species? This was actually a homework problem. So does somebody want to tell me how many chemical species we have here? Elmer. 6.
I think this was not the homework that they were at. 4. No, 6. Yes. 4.
There are 4 species. Right. Like the question in the homework was how many parameters do I have? So I should be careful. The homework had a variety of very similar reactions. And asked you to do more or less the same thing. And so we got, as I say, we got a little bit behind, in other words, in the lectures, so that I had assumed that you would have seen this particular derivation before you tried the homework problem. Yeah, Elmer.
What is it? Is it a complex ES, for example, an old species or not?
Well, we're calling it a species. We're saying ES, the complex ES is a chemical species.
It has the same variable character of everybody else. So it's not different mathematically. Right.
We have an amount of ES. Whether it's covalently bonded or whether it's reversibly bonded, van der Waals or something else isn't something that we can say. But mathematically, it's identified as a species. But that's a great point, Elmer. Good. Again, we always have this little bit of a tension between the underlying physical or chemical reality and the mathematics we're using. And so it's good to bring up these things. I appreciate it because when you do this a lot, it feels very natural, but it's not instantaneously natural. And I do still remember when I started doing this, it did not feel natural. So don't be shy about that. So we have 4 species, and so we have to write a rate law for each one. And we're going to do this systematically, and let's just look at it together. What I'd like you to do is start with the enzyme one and just walk your way through. I'm going to give people a minute or two. Just walk your way through how many arrows point to an enzyme, Either point towards or away from the enzyme.
The enzyme is ES.
No, the enzyme is just E. 3, yes. So that means there are going to be 3 terms in your equation for E. How many point to S? 2. So they're going to be 2 terms. So this is another sanity check. It's really easy when you start with a diagram like that to leave a term out. And so having a quick sort of counting the fingers on your hand and are they all there approach to checking is not a bad thing. ES is the how many arrows? 3. And what about P? 1. Good. So maybe just take a 30 seconds, 60 seconds and just walk through and look at E. And if E is on the source side of an arrow, you're going to get a minus whatever the value, whatever the rate sitting on top of the arrow is. If the arrow points to E, you'll get a plus with whatever the rate sitting on the arrow is. So see if you can just take a look and walk through and build for yourself, looking at the chemical reaction diagram, build those ODEs. You don't have to do all of them. I'll give you 2 minutes to just walk through and see where all those terms come from. And again, if I'm doing this by hand, I'll just walk through systematically. I'll start out E plus S. E plus S goes to ES. That means for my E thing, I'm losing 1 unit of E. And I look at the top of the arrow, I see KF E times S. So I write minus KF E times S. I just do that systematically. And one thing you'll see, is, of course, is that those chemical reaction diagrams are pretty compact. The ODEs take up a lot more real estate to say the same thing. William asks, why is the rate of forward reaction KF times E times S rather than KF times E plus S? And at some level, I could answer that. We're assuming the law of mass action, which says that is what it is. But there's also a sanity check on it too, which is if the rate of reaction were KF times E plus S and E were 0, I would still have a reaction if S were there, right? I want something that goes to 0 if either E is 0 or S is 0. Because I don't want my amount of E. I can't have my reaction happen if I don't have E, or if I need both E and S. So Elmer was talking about logic gates, and if you're coming from computing, multiplication is like an and addition is like an or. And so I can't have a chemical reaction of E and S if I don't have both E and S. So it's an and. So I expect a multiplication. That will seem extremely backwards to somebody coming from chemistry, but if you come from computing, it's sort of it may be a mnemonic to help you think about it. If you're really a mathematician, you'd say a Taylor expansion. The lowest term of the Taylor expansion has to be E times S. I have in the supplemental material for last lecture, the derivation of mass action. You can do it that way. There's some things that are so fundamental you can derive them from a lot of different points of view. You can derive it from a chemistry point of view. You can derive it from a probabilistic point of view. You can derive it from a mathematical point of view. You get mass action from any of those points of view. But the way you argue it is different. It's like proving the Pythagorean theorem. There are many ways of proving the same.
Oh, the most convincing one will be by modeling the probability of collision.
Right. Well, that's the physics approach, which is if I want 2 molecules to be within some distance of each other so they can react, then the probability of that happening depends on the density of either one. And And that's where that's the basic derivation. The reason it becomes a little more complicated again is that, and the reason mass action doesn't really work perfectly is that the molecules have a finite size. And so the probabilities are not quite what mass action says they are. It's assuming the densities are low.
The probability is so we are underestimating the probability of collision using mass action.
Well, it's a little bit more complex. Unfortunately, it becomes so in general, you're overestimating the probability. But it's not so simple. I mean,
because not every collision will provoke a reaction, right?
Well, so I should be repeating things for the people online. So Pedro is asking questions about the rules, what the assumptions are that need to be made for mass action to be true. And one of the things that has to be true is that the fraction of the volume occupied by the given species has to be very small. Otherwise, there are corrections and the actual reaction rate is lower than you would expect. There are some additional complications due to things like entropy, which can have an effect. And if the molecular sizes are very different, so you have a very big molecule like DNA and then a small molecule like a transcription factor, you can have what are called depletion forces that could actually give you faster rates of reaction than you'd calculate here. Mass action is almost never exactly right, but it's a pretty good first guess. In a lot of situations, it works really well. And so if you really don't know anything about chemical reactions, starting with mass action is unlikely to get you very far wrong. If you have beautiful experimental data, you probably have to correct mass action, or you know that the densities are very high. But in general, mass action works pretty well. Okay, so let's actually, before we run out of time, let's actually write a little simulation of this. So if people could fire up Tellurium and write this out. This was just an illustration of how a reminder of the syntax you need. Instead of J1 and J2, you'd have more lines, but they'd look similar.
Well, you don't even need that, right?
You mean the names?
Yeah.
Yeah, you don't need to name your reactions. I tend to always name my reactions because you never know when you'll need them.
Yeah, but will it recognize that you might have E plus S goes to ES?
Yes.
Times S or the ES?
No, it's fine. It'll take ES as a variable. Again, I think one of the great strengths of Antimony is that the way you type it is almost exactly the way it looks on the screen. The one thing you do have to remember is that if it's a bidirectional arrow, you'll have two lines, one for each arrow. You'll have one line for the forward arrow and one line for the backward arrow.
So then how would you, is there a fast way to assign multiple variables the same value?
Just equal signs. K equals K, K1 equals K2 equals K3. I tend always to write those out because it's too easy to make a mistake, but if you want to put them together, that's fine. And the one reason that you might want to make them separate is just because we may want to be able to change them individually later on.
They combined it work.
Putting them in sequence didn't work.
Yeah, well, I didn't use semicolons.
Well, then I just write them out. Like that. Is it working for people, not working for people?
Yeah.
What did you get? That looks good to me. How's it going for people? See if it works. I have a syntax there. See what I have to do.
You wouldn't do like,
Oh, I forgot to.
do parameter fitting on this, would you?
You could.
Cool.
We'll definitely get to parameter fitting a little bit later the semester. Did people get something like that? So this is the first, I would say, non-trivial result we have in the class, the enzymatic reaction. And you'll notice the key thing to look at in this one is there are a couple of things you could notice. First, this is an equilibrium reaction. All of the S eventually turns into product. Because there's no, once you create a product, there's no way back. So this is an ultimately irreversible reaction. So all the substrate eventually gets turned into product. The second thing to notice is the amount of complex, ES. It peaks quite early on and then begins to go down with time. That's the green line here. And you'll notice that E plus ES is a constant. So the amount of free enzyme goes down just as the amount of complex goes up, because the sum of those two is constant. And then the only other thing that you might notice is the rate of production of product, which is, if you look at the very beginning, the rate of production of product is very slow. There's this little flat bit right at the beginning of the reaction, because the complex takes time to form. Yeah, William.
Well, you said that notice how E plus ES is constant.
Mhm.
I mean, you can see that, but you can't be 100%. You mean that, like, just looking at the graph, you can't be 100% sure that it's constant. Right?
That's true. Absolutely. So
you graph E plus, yeah.
So William says, how do we show that it's actually constant? So there's actually till Antimony let us do that. If I define a new variable, I can define a variable which is E total, and I have to use colon equals. This is one of these slightly strange things. So I have to be. Somebody had the brilliant idea on this computer of having the keys, the characters on the keys flipped. So the top row is without shift, and the bottom row is with shift. And I mean, every other keyboard in the world is the other way. And of course, for the number keys, I know that. But for keys like semicolon, I actually look at the key to remember which is which. And so I'm continually typing the wrong equal sign instead of plus. I don't know whose idea it was to flip the keys. Somebody thought it was ergonomic to do this, but I'm always mistyping things. Anyway, semicolon equals will give you that. And if I now do E plus ES, it will now actually display the sum. Yips, well, I have to tell it to do it, I guess. Why is it not giving me that? Should tell me. It should have added to the. I apologize. I'd have to actually tell it to plot, explicitly plot E total. And the next exercise was going to show you how to tell it what to plot. And we're not going to get there today. I apologize, but that's how you do it. E total. If I say E total equals E plus ES, yeah, I can do arithmetic. Why do I have to use colon equals? The answer is regular equal sign is only evaluated when you load the thing. And so if I want it to be updated, I have to do that colon equals. But that's a great point. Don't trust. I said that they should be equal, but in fact, and the math tells me that they should be equal, but the computing could be giving me something wrong. So that's a very good kind of sanity check to do. Absolutely. Hattie, are you okay with that now? Is it working? Okay. Okay, so we've got something 5 minutes left. I told you I have a bunch of teeny exercises. So try S0 equals 10 instead of S0 equals 1. See what happens. This is called an enzyme-limited reaction. Now I've got lots of substrate and only a small amount of enzyme, which is often the case. In fact, it's much more common biologically that you have a small amount of enzyme and lots of substrate. How does it change? Does it change? So to do that, you only have to change one number. You have to put an extra 0 in front of the 1 for the S initial amount of S. Did it change the time series? You'll notice here, effectively over the time we're looking at, if your time, well, it depends a little bit on how long. If you run it long enough, you'll still run out of substrate. But if you run it for a short amount of time, now the amount of product will go up more or less linearly in time, because this is like using up the oxygen in this room. If I'm breathing, the amount of oxygen in the room is going down a tiny bit. But there's so much in here, basically, the amount of CO2 I'm putting out is essentially constant in time. I'm not limited by the amount of oxygen in the room. And so in this case, again, you'll see a little bit of a lag at the beginning while the complex forms. And then the amount of substrate will go down almost linearly in time. And the amount of product will go up almost linearly in time. And the amount of complex ES will be almost constant. In reality, of course, the amount of substrate is going down, and so the amount of complex will go down, and the slope of the product production will go down too. But over a short amount of time, these things are nearly linear. Did people see that? Okay, so you can already see two very different regimes with this very simple reaction. Another one is called quasi-steady-state. In quasi-steady-state, the rate at which the enzyme produces the product is very slow. And so now try this, take S naught S back to 1. Your initial value of S should be 1. And now make R, instead of making R 1, make it very slow, 0.01. What happens? Right, so here you get a lot of complex, and the amount of product is increasing. That's that red line at the bottom, but it's increasing very slowly, but it's linear, very linear. So if I didn't know this was a complex reaction, I'd just say, I'm producing this, I'm slowly producing product at a constant rate. So that's a pretty interesting one too. That's called quasi-steady-state, because things seem to be almost not changing. Now, suppose that I really want something to not change. I can do that by putting in a $sign. And if I put a $sign in front of one of the S's, and it doesn't matter which S I use, then S will be forced to keep its initial value. So now a lot of the math you've done isn't going to work anymore. But you could just try setting S initial value of S to be 10. And then put a $sign in front of the S somewhere. Tell me what happens. Now S won't change. you're forcing S to be constant. You're basically assuming there's an infinite amount of S. Did it work? Did it crash? Worked? Okay. So this looks almost like what we had before. And you'll notice in your plot something funny happens. S disappeared from your plot, didn't it? So just the way when I tried to answer William's question about what's the total amount, sometimes Tellurium gets too smart for its own good. If something doesn't change in time, it says it's not a variable and then it doesn't plot it. Now I can tell it, if I tell it explicitly plot this, this, and this, it knows the value. And so if I order it to plot it, it will plot a constant. But if it's a constant, it doesn't. If I don't tell it, or if I don't order it to plot it, it will say it's boring. It will hide it from you. I hope that was a good mix. I really appreciated people's presentations, and I hope that we had a good a balance today between doing a little coding, a little math, a little coding, some biology talking. So I want to thank everybody.